Hi class, today I'm going to be talking about the processes of mitosis and meiosis, which are both means of reproduction. Did you know that all of the cells in your body are constantly dying out? Well, how do we consistently have enough cells in our bodies, you might ask? Well, that is where mitosis and meiosis come in. They are parts of the cell cycle, which consists of an interphase stage and the mitotic phase. So we're going to start with talking about mitosis, which is part of the mitotic phase in most single-celled organisms and many multicellular organisms. Cells spend most of their time in interphase, where they go through a G1 phase, an S phase, and a G2 phase, where the cell is basically growing and DNA is being synthesized. During the mitotic phase, we have mitosis and cytokinesis. So during mitosis, there are quite a few stages. There is prophase, prometaphase, metaphase, anaphase, and telophase. And those all have to happen before the cytokinesis takes place. And here are some pictures of what each of these stages look like. So let's start right in with prophase. So during prophase, the nuclear envelope and other organelles inside the cell begin to break down and disperse, and the centrosomes move to the opposite ends or poles of the cell. And also the sister chromatids that are bonded together and make up the DNA are bonded more tightly with condensin proteins. In prometaphase, these steps carry on, and kinetochores are formed on the center of the sister chromatids. And this is also the stage where from the centromeres, microtubules come out, and some of these will attach to the kinetochores. During the metaphase stage, pairs of sister chromatids align in the middle of the cell in the area called the metaphase plate. During anaphase, these sister chromatids are pulled apart from each other and are pulled towards opposite poles of the cell, and they are pulled in the direction that they were previously facing. Telophase is when the sister chromatids, which are now individual chromosomes, begin to unravel, and on each side of the cell there are groups of chromosomes, and so on each side a nuclear envelope begins to form. So the second part of the mitotic phase, which we mentioned earlier, is cytokinesis. And in cytokinesis, which literally means cell motion, the cell is separated into two separate cells. And these two separate cells are an exact imprint of the parent cell, and they are two identical sister cells. This is different from meiosis, because meiosis produces a cell that is different from the parent cell or cells. So let's talk about meiosis. So meiosis happens with most multicellular organisms, but it also happens with a lot of unicellular organisms. Meiosis is more complicated than mitosis, but it still is preceded by an interphase stage. Meiosis is so complicated that it's divided into two stages, meiosis 1 and meiosis 2. So let's just start with meiosis 1. Meiosis 1 begins with a prophase 1, which is more complicated than the prophase in mitosis, because here it involves homologous pairs of chromosomes, and each of these chromosomes are made up of two sister chromatids, and they are tightly bound together by proteins. So after they are tightly bound together, there is a process where DNA is shared by the crossing over so the sister chromatids are going to cross over a non-sister chromatid, and they're going to cross over at chiasmata, which are specific points where they do this, and this is a way that causes variation because they are crossing over non-sister chromatids. And so this is a little confusing, so a picture might help to explain this. So we have the homologous chromosomes aligned, and then they cross over, and see these are the two sister ones, and then these are also two sister chromatids, so they cross over a non-sister chromatid, and then they end up creating some DNA that is crossed over there. So that is just a brief overview of the process of prophase 1. And so next we're going to have the prometaphase stage. So tetrads, which are just the combinations of 4 sister chromatids, were formed in that prophase 1 stage. So now in the prometaphase stage, as you may have guessed, microtubules will attach to those kinetochores. But then what we have now is you have those tetrads made up of a pair of chromosomes, and each of those chromosomes will face opposite poles of the cell. So next we have the metaphase stage, where they're all aligned in the middle of the cell, but they're aligned randomly, which is another source for variation. And then next we have anaphase 1. And during anaphase 1, we have the sister chromatids are going to stay connected together, but the separate chromosomes are going to go to the opposite poles of the cell. And then lastly we have the telophase, where these are going to start to unravel, and this leads to the cytokinesis, where they actually separate, and then you have 2 haploid cells, which are called haploid cells because they have half of the necessary DNA that they need. So after this, we're going to go to meiosis 2. And meiosis 2 starts with prophase 2, prometaphase 2, metaphase 2. These are all going to have the same sort of processes that we had in mitosis and meiosis 1. They had all these similar processes. So basically, by now, you pretty much know what is going to go on in these 3 sections. But after that, we lead to the anaphase 2 stage. And this is where we have the chromosomes, and the sister chromatids are actually going to separate this time. So we're having those sister chromatids separating, going to opposite sides of the cell, and they're going to go to those poles. And then during the telophase stage, they're going to, those sister chromatids are now called chromosomes because they're on their own now, and they're going to begin to decondense. And then of course during the cytokinesis step, that's when these cells are going to split apart. And so remember, we had the 2 haploid cells, and now each of those are going through this process. So now we're left with 4 haploid cells. So the result of meiosis will be these 4 haploid cells. And whereas the mitosis produced 2 identical sister cells that were identical, or most of the time they'd be identical to the parent cell, in meiosis we end up with 4 haploid cells. And so these cells are not going to be identical to the parent cell. So that is the difference between mitosis and meiosis. So thank you for watching. I hope you learned a lot today.